Okay, the recording started. So what I'd like to do is show you one of the most valuable resources you will ever have an opportunity to find. This is called Online Mendelian Inheritance in Man, is the name of the website. They will always ask you for a donation to start. If you are independently wealthy and have lots of money, go ahead and give it to them. Although if you're independently wealthy and have lots of money, call me. I have some ideas. Basically, Online Mendelian Inheritance in Man was begun by Dr. Victor McKusick, who was head of genetics, the genetics department at Johns Hopkins University. He had done stellar work in the 1950s evaluating bone diseases and cartilage diseases in the Amish populations in southeastern Pennsylvania, and had done a tremendous amount of work identifying the inheritance patterns of these diseases. And then over the years, he began to become more broadly interested in genetic disorders in all in human beings. And he began to keep track of them. I first got a copy of his Online Mendelian Inheritance in Man, which was a database that he put together back in the days of dot matrix printers. And in fact, it was in the early 80s that I got my first copy, and it was basically it was a dot matrix printout of genes and references and modes of inheritance for genetic disorders. The original printout was about maybe 1.5 inches thick in paper. In the late 80s, I got a copy of a more recent edition of his Mendelian Inheritance in Man. Obviously, it wasn't online. It was still in a book format. The book had grown. It was about between 3 and 4 inches thick. It was on it was bound in green cardboard and was basically still a bound copy of a dot matrix printout of a database format. Subsequently, in the 90s, Johns Hopkins was awarded a NIH grant to take all of the information that McKusick had assembled and put it online in an online database. Currently, it is part of the National Human Genome Research Institute. That's that NHGRI. We'll talk about them in just a little bit. And it is available for you. It's available for you free, except for that little advertisement up front asking you to donate money. And you can pretty much go in here and look for any genes of interest. Folks who are taking my genetics lab know that one of the last, and everybody else in genetics lab know that one of the last exercises in lab is to draw family pedigree. However, in my lab, I require that everybody look up a trait on Online Mendelian Inheritance in Man to find out if it really is inherited in a Mendelian fashion, and if not, if it is inherited at all. So this is a huge resource. However, I'm not going to show you. You can always go in here. It's fairly easy to use. There's a search bar in the middle. You can just put in whatever trait you want, and it will come up with a series of answers. But what I really want to show you are the statistics, and I find these to be absolutely fascinating. You can see pretty much why the database is online at this point in time. And if we just look at the top row, we've got the gene description. Okay, We know that genes can be inherited autosomally, X-linked, Y-linked, or in eukaryotic cells, the other way that genes can be inherited is through mitochondria, because remember, mitochondria have their own bit of DNA, probably ancestrally related to their prokaryotic ancestors. Okay, so we've got autosoma, X-linked, Y-linked mitochondria, leading us to about 16,500. Again, I'm rounding. I can read it. 16,500 different descriptors of genes. These are all in human, and they range from simple phenotypes to more complicated phenotypes. What do I mean? Simple phenotypes, hair color, mid-digital hairs, polydactyly, achondroplasia or dyschondroplasias. Okay, these are defects in cartilage formation and ultimately bone formation. Some of these are very simply inherited. Then, of course, we have the more complicated patterns of inheritance. For example, skin color, hair color, eye color. Colors are always more complicated in terms of inheritance than some of the monogenic, meaning single gene phenotypes. And so 16,500. To me, what is absolutely striking about this is you take a look down at the third row. And this is where the phenotype, meaning the Mendelian phenotype or disorder, where the molecular basis is known. Again, in each of the columns, but let's just look at the total. 6,000 out of 16,500, where we understand the relationship between the biochemistry of the gene and the actual phenotype. Now, you may think, well, gee, that's really not very much progress. In reality, it's phenomenal progress. But what about the other 10,000? What about the other 10,000 phenotypes that we really do not understand the molecular basis of? Is it because people are not looking? The answer is no. People are always looking. Everybody's looking for the molecular basis for all these phenotypes. The difficulty is if we think about, remember way back when in our first lecture, I said, what are some definitions of a gene? And the obvious phenotypes that you get from your parents. Why do you look like your parents? Okay, simple inheritance. The more complicated definitions of a gene were a piece of DNA that codes for a protein, more along the lines of central dogma, right? More along the lines of central dogma. When we think about central dogma, piece of DNA that is transcribed into RNA and a piece of RNA that's translated into a protein. That actually is a very simple idea of what a gene is, and probably accounts for over 5,000 of those known 6,000 genes that have a molecular basis that is identified. What this suggests to us is that those 10,000 other genes that are inherited in a clearly identifiable manner by using pedigree analysis are not simply explained with the ideas underlying the concept of central dogma. This is really striking. this is really striking because it means that genetics is way more complicated than we really understand by using the concepts of central dogma. Now, remember, I said about a little over 5,000 of those 6,000. So what is encompassed in those couple of thousand other, or rather those about 1,000 other phenotypes where the molecular basis is known? Well, these are genetic concepts that are not accounted for by central dogma. The simplest of those are just RNA splicing variants. That is, we know that eukaryotic genes have both introns and exons. The exons are removed, enzymatically excised, okay, excised, or rather, sorry, the intervening sequences are removed and the exons are spliced together in order to eventually create the messenger RNA that will be translated into a protein. But sometimes during the splicing of the exons, they're not spliced in the correct order. And so we can actually have multiple gene products from a single DNA sequence. All right, so you remember this from the readings and thinking about messenger RNA and the structure of the eukaryotic gene. So we've got some of those, but it turns out that there are many more complicated events that occur to account for those 10,000 other phenotypes that are clearly inherited from parent to offspring, but cannot be explained by what we currently know about the molecular biology of the gene. So very often, especially in younger students, I usually encounter this with folks either right out of high school or certainly in freshman biology, and you start thinking about central dogma. And once concepts of central dogma are approached and learned, everybody says, well, genetics is over. We already know everything we need about it. We know the structure of DNA. We know how it replicates. We understand how it gets converted to DNA, gets converted to messenger RNA. We know that messenger RNA is translated to a protein. We know that there's some post-translational modifications. Sometimes proteins get chopped up and they get enzymatically processed to create some other product. And so genetics is done. We understand everything there is to know about genetics. But the reality is we probably at this point in time know only about a third of everything we need to know about genetics. We are not close to being finished. So I want you to realize that as much as we've discussed over the course of the semester, there's a long way to go. And there's a lot we don't know beyond this. So what I want to show you are some tools that will enable you to begin to attempt to answer some of these questions. That is, how can we begin to evaluate and understand what's going on with those other 10,000 genes that we don't understand? 10,000 phenotypes inherited in a Mendelian fashion. So when I say gene in that context, and again, the vocabulary in genetics is very important. When I say genes, these are phenotypes inherited in a Mendelian fashion, but we still do not understand the molecular basis of them. And there are a lot of really interesting ones. So I want to show you some tools that'll help you to get to the point where you understand them. One of the places that most people begin is here. Sorry about that. Is here. This is the National Center for Biotechnology Information. It is a tremendous resource. A lot of people use this as their entry into trying to understand the details of inheritance and the molecular basis of genes. Notice that it has a very simple entry point. We get to the top and we see that it has a search bar that allows you to probe all their databases. I want to show you their databases. And notice the huge number of databases that are now available. Right in the middle, by the way, is OMIM. There's our online Mendelian inheritance in man. So you can search Dr. McKusick's original database, and actually all the things that have been added to it since he passed, directly from the NCBI site. And this is why it's important for you to know where this is. Some of you in some of your other classes and projects may have seen PubMed. This is where you can get access to searching for all of the referee journal publications that are regarding usually medically associated. PubMed used to be actually only a medical information directly related to human medicine, and there was a separate database that was searched for animal or agriculture or plant areas of interest. However, that they have now all been coordinated into PubMed. So even though it still has the abbreviation of medical publications, it is not specifically tied directly to human medicine any longer. It's pretty much everything you can imagine that's published in science. There are still a handful of journals that are not summarized in PubMed, and usually you can get to them through the library resources. But pretty much everything you need is in PubMed. I'll show you one other one you actually probably know. Things that you cannot find in PubMed these days are really easy to find in Google Scholar. And as much as I hate to advertise for the evil empire that is the Google empire, they're nearly as bad as Microsoft these days. The Google Scholar is a tremendous resource. It covers pretty much everything that is found in PubMed, but even goes beyond PubMed. It also has recommended articles that are based on your prior viewing and your prior searches. And so, of course, what you can see here is a reflection of my interest in genes that influence fertility. And specifically, I have been long interested in, and if you ever looked up my publication record, you'll see that I'm interested in genes that are involved in producing proteins that allow sperm to bind to oocytes just prior to fertilization events. Sounds like a rather narrow specialty, doesn't it? I mean, we're really focusing in on a handful of genes. But there's some really interesting mutations we've discovered along the way. And obviously, they're tremendous economic importance as well as human reproduction importance. So you can kind of see some of my background materials here as well. So Google Scholar, awfully handy, especially if you're interested in things that are not covered in PubMed. However, PubMed is probably the default. A lot of people, I've noticed a lot of people at Stockton, you may have had other professors send you to PubMed. It's just the normal go-to reference. Also on NCBI, you can look at this left-hand column and notice that there are a tremendous set of subchapters and pages. You can look up not just organisms or references. You can find chemicals, techniques known as bioassays. You can identify data sources. So there are free, publicly available data sources that you can download. So for example, if you're interested in cancer incidence in the US or in worldwide in different types of cancer, you can get to the publicly available cancer databases through this. You can also get software that is freely available for you to download, free, love free, freely available for you to download onto your computer to use. And there are a handful of really great software packages that as you progress in science, you'll probably have an option and may have an opportunity to use in your work. There are specifically DNA and RNA databases, genes and genetics in medicine, genome maps, a big part of human genome maps. You can directly, literature is mostly PubMed. You can also do sequence analysis, taxonomy. Their trainings and tutorial sections are fantastic. They have videos, again, free videos that you can take a series of courses and actually learn how to use all of the resources on NCBI. A lot of really top-notch resources here, all in one place. Personally, I find the NCBI site boring. It's really not visually enticing. I find sometimes it's a little bit difficult to identify the tools that you want to use, although you can notice if you are interested in COVID these days, they have a number of great links right up top where you can learn quite a bit about the CDC recommendations for public health, some of the research information. You can actually get the public data for the vaccines here. SARS-CoV-2, you all know that the official name of COVID-19 is really considered to be the disease, but the virus that calls it is part of the SARS coronavirus 2 virus. It is the specific name of the virus. You can get sequence information. I know that Dr. Luke in her bioinformatics class this year is having the students do some independent projects looking at the DNA and protein sequences that are coded for by SARS-CoV-2. So really some fun stuff. What do you want to know about prevention and treatment? And obviously it is also available in Spanish. So this is someplace that you can poke along if you have projects that you're working on. Great place to start to get some information. But I'd like to show you my go-to resource for biotechnology information. And it's a little place called Expasy, EXPASY. And it is run by the Swiss Bioinformatics group. It is actually part of the European Molecular Biology Group or Biology Labs, which is known as EMBL, E-M-B-L, European Molecular Biology Labs. And I find this to be actually, I use Expasy almost exclusively compared to NCBI, and you'll see why in just a little bit. What are some of the tools that they have available? And you can see on the left-hand side, I really do like their organization structure: genes and genomes, proteins and proteomes, evolutionary and population biology, structural biology. A lot of fun in the structural biology section, allowing you to create 3D structures from protein sequences, also systems biology, and then of course, the newest topic in genetics and molecular biology, which have the underlying concepts of data mining and machine learning. And this is really the recognition that genome sizes are big, okay? Think about the size of the human genome, all right? About 4.5 gigabases, okay? 4.5 gigabases of information that result in approximately 20 to 20, about 22, but I always say 20 just as shorthand, about 20,000 different genes. And if you're interested in looking for particular sequences or understanding the organization of those genes, and certainly in humans, we're talking 23 pairs of chromosomes, just mathematically, it is a tremendously difficult problem to conceptualize in your head. So some of the techniques of using machine learning to answer questions and having the machine go through and run simulations and look for patterns in these very, very large numbers, really in a very exciting area right now in genetics and molecular biology. Just think about it like this. Everybody here should be, by reflex, able to say, well, if we have a monohybrid cross with a dominant and recessive allele, what's our expectation in the F2 generation for distribution of the progeny? Everybody here, immediately. Monohybrid cross, right? Crossed two hybrids, cap A small a, right? Cross the two hybrids, you get progeny, and our expected phenotypic ratio, 3:1, of course. I know most of you know this. And the genotypic ratio, 1 to 2 to 1. Right? Piece of cake. If we go to a dihybrid F2 population, so when we're thinking dihybrid, cap A small a, cap B small b, crossed with cap A small a, cap B small b, okay? Classic F2 dihybrid cross. Both genes have a dominant and recessive allele. Nice and easy. The phenotypic ratio, everybody here knows, by reflex, right? Expected phenotypic ratio. Give you a minute to think. I lied. Give you 5 seconds to think. Everybody knows 9 to 3 to 3 to 1. Piece of cake. Perhaps less obvious for you, although something in your mind. What's the genotypic ratio of an F2 dihybrid cross? Phenotypic, 9 to 3 to 3 to 1. Genotypic, 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. If you were adding along the way, you know that's a total of 16, so we know that that's correct. All right? So obviously, I don't expect you to memorize the genotypic ratios, but in your mind, it's really not that hard, meaning the human brain is capable of doing this. But now, let's take two human beings from completely different areas of the world whose populations have never inbred before. So I don't know, someone from the deepest, most remote areas of a South Pacific island, of islanders who were in one place and had never before been encountered by other human beings. And then perhaps another group of Aborigines from perhaps Australia that have never really encountered or certainly been introduced to more modern populations. And those two individuals being more or less homozygous meet. Maybe the aliens came down, beamed them up, they met on the ship and they got together, they fell in love and now they're gonna have kids. So now we've got two individuals, each having 20,000 genes. So our Punnett square, instead of being 4x4 for our F2 dihybrid, for our F2 20K hybrid is going to be 40,000 by 40,000 unit Punnett square. We don't have a hope. Okay, there's no way anybody is able to think about this. However, we do have computers that are able to compute the odds and compute the total number of possible genotypes. And so how do we decide what's important and what's not important? And the answer is our minds are not capable of actually evaluating a mathematical problem that large. So what do? Machine learning. Really exciting area, artificial intelligence, really exciting area right now in genetics and molecular biology. So some fun stuff here. So I have a little project that I'd like to share with you. And I am going to, let's see, just one second. Let me get, I'm going to share a slightly different screen with you. Just a moment. I'm on my Mac. It takes me a little bit longer to, there we go. It takes me a little bit longer to maneuver around my Mac. Anybody ever see the movie Jurassic Park? Or any of the, I don't know, 15 or so sequels that were in Jurassic Park? Everybody's watched Jurassic Park. No? Oh my God, go, well, read the book. It was better anyway. Go read the book. Jurassic Park, if you recall, even the story, it's a classic story by Michael Crichton. Michael Crichton wrote this book in the 80s. And when the book came out, it was phenomenally exciting. Basically, someone was able to get some DNA from dinosaurs. The way that they found the dinosaur DNA was they found mosquitoes that were captured in amber. And of course, because mosquitoes feed on blood of other creatures, they were able to extract the blood from the mosquitoes captured in amber. And of course, if the amber is old enough, whose blood is in the mosquito? Dinosaur blood. Okay, so they got the dinosaur blood, they sequenced the dinosaur DNA, they took the dinosaur DNA, put it into turkey eggs, because dinosaurs won't fit inside chicken eggs. They put them inside turkey eggs and some of them ostrich eggs, and grew them out and hatched them, and they created dinosaurs by using modern molecular and reproductive technologies. And in Michael Crichton's book, he actually has two pages in the paperback at any rate. He has 2 pages of what you see here on page 103 of a DNA sequence. Okay, so here is the DNA sequence. And it was really kind of exciting for those of us, you know, who were involved in genetics at the time, to be able to actually see a string of letters, ATGC, ATGC, all of the different ones, keep going, keep going, keep going, in a New York Times best-selling book. And we were like, wow, you know, genetics is here. People would say, well, what are all those letters? And we got a chance to talk about it in class. And in the 80s, were when many of the tools were developed that allowed us to begin, because remember, the Human Genome Project started in 1981. And so we really began to look at this and say, I wonder what the heck he did. Most of us at the time the book came out assumed that all he did was kind of randomly type 4 letters over and over and over again, and so he could put it in his book. But wouldn't it be interesting if we were to find out what protein this really is? So I could put this on the final exam, and I could give you a codon usage table and say, figure out what protein this is. What do you think? Fair? Oh my God. You'd be taking the final until next semester. Okay, obviously unfair. However, I'm going to take the sequence and I'm going to copy it. I'm going to copy it. Now we don't need to look at that anymore, but we're going to go back to our handy dandy XBASE. And in XBASE, we've got some really unique sets of tools. We've got unique sets of tools. One of the most powerful tools in the box is to look at the Genomics Tools. And I'm just going to click the upper box so we could take a peek. And notice all of the different tools that are available to look for and to probe for sequences, okay? I'm going to show you an easy one. And we are going to go, my screen is hiding out on me here. We're going to look up some really interesting, in fact, I'm just going to search for it up here. Here you go. We've got, here's our database called Translate. And all we have to do is go into the Translate tool, paste our sequence, I actually kind of like to use this. It's called the verbose format. And the reason is I like my methionines to stand out and I like my stop codons to stand out. It's easier for my old eyes to identify than to look for M's and dashes. And hit the button Translate. And instead of having to sit around with your tool to translate the DNA sequence into a protein sequence, it automatically puts together the sequence into the DNA sequence, automatically converts it for you into protein sequence. Now, one of the fun things here is notice that it also doesn't know whether the single-stranded DNA code that I gave it is read in the forward direction. It doesn't know if it's read, meaning the 5' to 3' direction or the 3' to 5' direction. It doesn't know where the gene begins and or ends. All I did was ask it to convert triplets of DNA into our individual protein sequences, okay? Which means that there are 6 total reading frames. So it does provide you with all the reading frames, reading frame 1, 2, and 3 in the 5' to 3' direction. And I'll show you 1, 2, and 3 in the 3' to 5' direction. So the tools available, and you could see the large number of methionines, really easy to see because we see the MET and stop codons that are available as well. So it doesn't entirely make a lot of sense in the context is it doesn't code for one single protein, okay? There's a lot of starts, a lot of stops. But you can also see why it's important for you to know the single letter code for all the amino acids. So you can go right in and start reading alanine, leucine, leucine, alanine, phenylalanine, phenylalanine, histidine, arginine, leucine, arginine, proline, proline, aspartate, glutamate, histidine, histidine, K is lysine, arginine. Okay, I'm not going to keep going. But it becomes very easy to read these if you know the single letter code. So tremendous utility of having the computer as a way to do our translation for us. And of course, this is about 1,000 base pairs. So if we have 1,000 base pairs, you can figure out very quickly, there are probably about 333 right amino acids, right? Because triplet codons code for a single amino acid. So if you have 1,000 base pairs, you divide by 3, and that's about 333. So you can convert very quickly from the number of DNA base pairs to the number of amino acids, and you can actually see the sequence of amino acids right in the translate database. So really very exciting spot to go. But that didn't really tell us a lot. It didn't really tell us a lot of information other than to be able to do the translation very, very quickly. So what if we had a way to find more information? And I'm going to go back to our NCBI. Oops, wait a minute. Sorry. Here we go. I'm going to go to another tool, and notice it's right up here at the very top. The tool is BLAST, okay? The tool is BLAST. And we're going to go, there are 2 of them that I want to show you. And we will go to the easiest one, which is BLAST, take us directly to the resource. And notice it's asking us for exactly the same thing. We are going to poke in, we are going to paste our DNA sequence right in there. Now notice on the right-hand side, there's some really interesting variants of BLAST. BLAST can be done for proteins, BLAST can be done for nucleotides, it can be converted, it can be, doesn't matter. And each of them use a slightly different algorithm. If you take bioinformatics, you'll learn what those differences are. But the bottom line is you don't really need to do much about this. Just plug it in here and run BLAST. And notice, instead of just giving us an output of the expected amino acid sequence, it's going to tell us, waiting, it's going to tell us, come on, it's going to tell us the origin of the sequence and how it compares to all reported sequences in the nucleotide and/or protein databases available by, publicly available at any rate. It's still thinking, hang on. Sometimes it takes a minute or so to run. BLAST was a software program originally created by Webb Miller. Webb was at Penn State when I was there, really a great colleague, a tremendous bioinformatician, a really great computer programmer too. So if you want to think about doing some computer program, these are the kind of places where we really need good minds to get in. It's still running. Making sure I didn't lose my internet connection again. We'll give it a moment. And just so you know, I tend to like Xpasy, but what I'm going to do while it's running, I'm just going to switch over. You can see pretty much if you were to look in NCBI, you can go directly off my screen here. You could see down here in the middle, there is the BLAST information also on NCBI. We're going to look here. It gives you the choice of actually looking at nucleotides. Let's see which one's faster. The nice thing about, there you go. So NCBI is clearly faster than Xpasy. We'll let that go. Now let's say, look at this. Sequences producing significant alignments. And if you take a look at it, the identity, and you can read the identity on the side, is 99% identity. That is the dinosaur sequence that was published by Michael Crichton is identical to, take a look at what they are. They are cloning vectors. And in fact, where he got it was this one right here. It is a plasmid from E. coli. So at the time he wrote the book, the only sequences that were available were sequences from prokaryotes. So what he wrote in the book was a sequence of dinosaur DNA, was actually a plasmid from E. coli. Was a plasmid from E. coli. So pretty funny little aside from the book, it turned out that he really was just looking for some kind of real DNA sequence, was disappointed when he called all the biochemists and geneticists of the time working on the Human Genome Project and discovered that nobody had any real sequences for him. So they said, yeah, but we have these full DNA sequences from E. coli. And he said, oh, okay. He said, I'll use that. Nobody will ever know the difference. Well, I'm happy to report that 40 years later, everybody knows. So good thing he's dead, because now he'd be very embarrassed to find out that we caught him. Now, a funny end to the story, I guess I find it a funny end to the story. A crazy end to the story is, of course, there's more than one Jurassic Park book, just like there's more than one Jurassic Park movie. And he published the second Jurassic Park book about 10 years later. And guess what? He put another DNA sequence in that book. And he said, oh, this is a very important gene controlling the growth of the dinosaurs to make sure that they don't get overly aggressive and kill all the humans. Okay? And so he published the sequence of the gene. Well, of course, by this time, all of us had these brand new tools that we could use to find out what gene it was. And it turned out that the only gene that they had available at that point in time that he used was he used the gene from chicken muscle. And it was actually, it was a muscle gene of actin, which is one of the parts of the muscle fiber in humans and in fact, all vertebrates. And he was able to take that sequence from the chicken. So in his first book, he had an E. coli gene and represented it as a dinosaur. In the second book, he had a chicken gene and represented it as being part of a dinosaur. And of course, for my money, that's why we know that dinosaurs still live today, except they're usually on our sandwiches. So they're not really part of, we're not so worried about being eaten by T-Rex anymore. We're the ones who do the eating. So if you get a chance, next time you're at Popeyes and you get yourself one of those nice hot chicken sandwiches, okay, just remember you're eating an ancestor of T-Rex. So turnabout's always fair play in these things. So I wanted to give you an opportunity to see that we're able to, there's actually something down the road from here, opportunity to see some of these databases. I hope that if you have the opportunity, whether in research projects later on during your time at Stockton or in other classes, that you have an opportunity to use some of these tools. They really are very valuable. I highly recommend, if you're going to be working on pedigrees in genetics lab, which is usually the last of the genetics labs, that you're able to use online Mendelian inheritance in man. It is a hugely valuable tool to be able to go in and see what the expectations are for genes that have been previously reported. And I do recommend that as you look into the different phenotypes on OMIM, that you also note whether or not the molecular basis of that particular phenotype is known, because there's really a 2/3 chance or so that whatever you're looking at is not known. These are things that we've yet to discover. And those two sites that we looked at, the National Center for Biotechnology Information and also the XPASY site from the European Molecular Biology Labs, are really two of the places that you'll find the tools that will enable you to parse out the molecular basis for inherited traits. So that's where I wanted to go today. I hope it was a little bit different, a little bit more fun than just going through some of our other materials. And looks like they changed the forecast. We're not going to have rain this week. So get to the beach. Go do something fun. All right. You guys take care. Any questions, answers? Going once? Cool. You guys have a great weekend. Talk to you later.
Have a great weekend, Professor.
See you.